So we're going to look at a worked example of codominance and incomplete dominance here. The difference between these two expressions, and actually you'll see the genetic cross is the same for both, but incomplete dominance is when you have blending of a colour. So you end up with a new colour like pink, which you didn't have before. And codominance is where both alleles show. And this is where maybe you have a coat colour that is a mixture of two colours. So it might be a colour we call blue, which has black and white, and they both show at the same time because it's actually a speckled mixture of some black and some white. And in the coat colour, because you've got some of each being expressed, you end up with a new option, but it hasn't actually blended within one cell. But when you come to do the cross, it's the same. So let's have a look at a flower where we have a red flower crossing with a white flower. Now we have a slightly different terminology that we use for when we've got incomplete or codominant, and it's not dominant and recessive. And the way I'm going to show it is by saying that the allele for red is represented by C for colour, but a superscript R. Now remember, we always have two alleles for a genotype. So if you have a red individual, it needs to be homozygous for the red allele. So that whole thing is the red allele. And then we have a genotype with two alleles for the individual. So the white flower is a big C for the gene for colour, but the allele is white. So gene for colour, allele is white. Now when we breed these together, we've got to work out what the gametes are. The gametes for this individual will all be colour white. And for this individual, their gametes will be colour red. So you can add a label for this line if you want to. That's the gametes. Let's put them into a Punnett square. The gametes from this individual, there's only one option. So we'll put both the gametes the same on this side. And from this individual, there's only one option again. All of the gametes are carrying red alleles. So every single one of these squares will be the same. Again, go alphabetical if you can. The red allele and the white allele, and that will be the same for all the boxes. So all 100% a new phenotype that we did not have before, which is pink. All of them are the same. Now what's going to happen if we take two of these and breed them together? Let's have a look. So lay this out properly. A parent phenotype is pink. So pink crossed with pink. The parent genotype, which is what the alleles actually consist of. So that's the colour gene with the red and the white alleles. And then the gametes, which we can actually put straight onto our Punnett square. So here is our Punnett square. The gametes from this individual are half of the gametes are going to contain a red allele, half of them will contain a white allele. Put the gametes in circles, making it clear to the examiner that you realise they're supposed to be on their own. That's a gamete now, not an individual. So remember to put them into circles. These are the gametes from the other parent, and in each square, combine through fertilization, bring those two gametes back together, and we'll make some new individuals. The first one is red. This one, go alphabetically, is pink. It's got a red and a white, and they're blending. This individual has got a red and a white, and they're blending. And the fourth square, we have a white allele coming from one parent and a white allele from another. So our third genotype is white. So now when we do our ratios, our phenotype ratios, we've actually got red and pink and white in a 1:2:1 ratio. And you'll notice here we have 3 phenotypes. That is our clue. That gives us a clue that it's going to be codominant or incompletely dominant. And when you lay out the cross for these two, the cross looks the same, just the terminology changes depending on what's happening in the phenotype, whether you're seeing both colours together on the same organism or both colours making a new colour in the cell.